There are a total of 6 distinct tastes available to us. According to recent research, the human tongue may be capable of identifying a 6th basic taste, in addition to the well-known flavors of sweet, salty, sour, bitter, and umami. This newly discovered taste is believed to be ammonium chloride. In the early 1900s, a Japanese scientist named Kikuna Ikeda proposed the concept of umami as a fundamental taste, on par with the familiar tastes of sweet, sour, salty, and bitter. It took approximately 80 years, but eventually the scientific community came to a consensus and recognized Ikeda's proposal as valid. Researchers at the USC Dornsife College of Letters, Arts and Sciences have uncovered evidence of a 6th fundamental taste, according to their findings. In a recent piece of research published in Nature Communications on October 10th, Emily Lyman, a neuroscientist at USC Dornsife, and her team discovered that the tongue reacts to ammonium chloride using the same protein receptor that is responsible for signaling the sensation of sour taste. Lyman, a professor of biological sciences, suggests that individuals living in Scandinavian countries may already be accustomed to and fond of this particular flavor. Salt licorice has been a beloved candy in certain northern European nations for over a century now. The candy's ingredients include salmiak salt, also known as ammonium chloride. For many years, scientists have acknowledged the tongue's heightened sensitivity to ammonium chloride. Nevertheless, even with copious amounts of research, the precise receptors on the tongue that react to this chemical remained shrouded in mystery. After much consideration, Lyman and the team of researchers believed that they had a potential solution. Recently, scientists have made a breakthrough discovery in identifying the protein responsible for detecting sour taste, known as OTOP1. This protein is found within the cell membranes and functions as a channel for hydrogen ions to enter the cell. Acids are primarily comprised of hydrogen ions, and it is common knowledge amongst gastronomes that the sensation of sourness is stimulated on the tongue by these same hydrogen ions. This is why lemonade, which is abundant in both citric and ascorbic acids, vinegar, which is composed of acetic acid, and other acidic foods tend to impart a tangy flavor sensation when consumed. The OTOP1 channel is responsible for the uptake of hydrogen ions from these acidic substances into the taste receptor cells. The team was curious if ammonium chloride could initiate OTOP1, as it has the ability to impact the acid concentration or hydrogen ions within a cell. In order to address this inquiry, the researchers introduced the OTOP1 gene into human cells that were grown in a lab, prompting these cells to generate the OTOP1 receptor protein. After this, the cells were subjected to acid or ammonium chloride, and the responses were measured and recorded. As per Lyman, it was observed that the activation of the OTOP1 channel is immensely augmented by ammonium chloride. This activation is found to be as good, if not better, than the activation caused by acids. The process of ammonium chloride results in the release of minimal quantities of ammonia. This ammonia then moves within the cell, elevating the pH level and consequently rendering it more alkaline. This alkalinity in turn leads to a reduction in the number of hydrogen ions present. Ziyu Liang, the primary author of the study, explained that the OTOP1 channel generates an inflow of protons due to a disparity in pH levels. In order to verify that their findings were not just a result of lab conditions, the researchers utilized a method that mimics the transmission of signals through nerves by measuring electrical conductivity. To accomplish this, they compared the responses of normal mouse taste bud cells to those of cells from mice that had been genetically altered to not produce OTOP1. The scientists observed the electrical responses, known as action potentials, generated by the taste cells when ammonium chloride was introduced to them. Upon the addition of ammonium chloride, wild-type mice's taste bud cells displayed a drastic surge in action potentials. Conversely, the taste bud cells from mice who lacked OTOP1 failed to elicit any response to the salt. This experimentation confirmed the hypothesis that OTOP1 is responsible for the response to salt, which in turn generates an electrical signal in taste bud cells. While studying the reactions of nerves that control taste cells, Courtney Wilson, a member of the research team, made a similar observation. Specifically, she found that the nerves responded to the introduction of ammonium chloride in regular mice, but not in those that lacked OTOP1. Afterwards, the research team decided to explore how mice would behave when faced with a choice between plain water and water infused with ammonium chloride. To conduct these tests, they disabled the bitter cells responsible for the taste of ammonium chloride. Mice with a functioning OTOP1 protein found the taste of ammonium chloride to be uninviting and did not partake of the solution. Conversely, mice lacking the OTOP1 protein showed no aversion to the alkaline salt, even at exceedingly high concentrations. Lyman expressed how crucial the OTOP1 channel is for the reaction to ammonium, stating that this was truly the deciding factor. The scientists continued their investigation, curious if additional animals possessed the capability to detect ammonium via their OTOP1 channels. Their research revealed that certain species exhibit a greater sensitivity to ammonium chloride through their OTOP1 channels in comparison to others. Astonishingly, the OTOP1 channels in humans also display sensitivity to ammonium chloride. The evolutionary significance and advantage of consuming ammonium chloride is a topic of inquiry. What purpose does it serve and why has it persisted over time? Lyman postulates that the capability to detect the taste of ammonium chloride could be an evolutionary development serving the purpose of preventing organisms from ingesting hazardous biological substances that have elevated levels of ammonium. According to the expert, ammonium can be found in various waste products such as fertilizers and is known to possess some degree of toxicity. This explains why our taste receptors have evolved to recognize the presence of ammonium. Interestingly, research has shown that chicken OTOP1 is considerably more sensitive to ammonium compared to zebrafish. These variations in sensitivity could be attributed to differences in the ecological habitats of these animals. For instance, fish may not encounter high levels of ammonium in water, while chicken coops are typically filled with ammonium that must be avoided and not consumed. While the research on sensitivity to ammonium is still in its early stages, caution must be taken. It is essential to understand the differences in sensitivity to ammonium among species and identify the factors that cause some OTOP1 channels to be more sensitive to ammonium than others. Further study is required to gain a more comprehensive understanding of this topic. In order to achieve their goal, they have taken the first step. Lyman explained that they have pinpointed a specific amino acid in the OTOP1 channel that is crucial for its reaction to ammonium. By altering this single residue, the channel's sensitivity to ammonium decreases, yet it still remains responsive to acid. According to the expert, this particular amino acid must have played a critical role in the survival of different species, given its conservation across these organisms. This implies that there was a selective pressure to maintain this amino acid. Specifically, the OTOP1 channel's capacity to react to ammonium must have been vital to the animal's survival. The scientists are intending to broaden their scope of study in the future, to determine whether the sensitivity to ammonium is preserved in other members of the OTOP proton family. These members are present in other regions of the body, such as the digestive system. It is possible that ammonium chloride may become recognized as a sixth fundamental taste, joining the current roster of 5 basic tastes. Only time will tell if this is indeed the case. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.